AS Level Biology 9700 Paper 1 Variant 2 October November 2025, and this is the second video on this paper, and we will discuss from question number 15 to 27. So first of all we will go with the question number 15, and question number 15 is: some students investigated the effect of pH on the rate of starch digestion by MLAs. At 20 degrees centigrade, they added 2 cubic centimeter of 1% MLAs and 1 cubic centimeter of pH buffer solution to a test tube. They added 2 cubic centimeter of starch solution containing 0.0 gram of starch to the test tube. They measured the time taken for the starch to be digested. The students then repeated these steps with pH 5, 6, 7, and 8 buffers. Now you can see the results in the table. At 5, at pH 4, it took 4 minutes, 5, 1.5, 1, and at 7, it is 2.5, and at 6. You can see as the pH is increasing, the time taken for starch is decreasing from 4 to 6, but at 7 again it is start increasing, and at 8 it is the highest time. 8 pH, the time is highest, so it means the rate of digestion of starch has been decreasing. Now we have to move towards the next page, in which they have shown us 4 graphs, and we have to choose only one. Which one is the correct answer? So you can see these graphs. These 4 in on x-axis of these graphs, you can see they have given us the pH value, but on the y-axis they have given us the rate of starch digestion, not the time for digestion. So first of all, we have to calculate the rate of starch digestion, and the rate of starch digestion is equal to 1 by time. So first of all, at pH 4, the time is 4 minutes. So we will divide 1 by 4. It will be equal to 0.25. And pH 5, the time is 1.5 minutes. So we will divide 1 by 1.5 with, and then it is equal to 0.67. And pH 6 times 1 minute. So we will divide it 1. So it will be equal to 1. At pH 7, the time was 2.5. So we will divide 1 by 2.5. It is equal to 0.40. And 8, it was 6 highest time. So 1 by 6 will be equal to 0.17. Now you need to understand that the highest rate of reaction is at 6, which has been given in minutes. So you can go with the graph B and C. They have given with the second. So B and C, both B and D both are not correct because they have given in seconds. Now we will go with the A and C. Now we have to choose the correct. Highest rate of reaction is at pH 6. So pH 6. So you can see the highest rate of reaction has been, which is highest, has been given at graph number C. And in A shows the lowest pH. It shows the lowest pH. So A is not the correct option. Now go with the C, and you can compare it with the values which we have calculated. At C, at 6, it is the highest rate of reaction, which is 1. And then the second highest rate of reaction is at pH 5. which is 0.6. So one value, second, and you can compare it with which I have calculated, and the third value goes at pH 7, which is 4, and the fourth one value is at 5, which is 0.6, and the fifth one is the lowest rate of reaction is at 8, which is 0.1. So we will go with the graph C. C is the correct option, which shows the highest rate of reaction, because they have calculated, they have drawn the graph between pH and rate of reaction, not between the pH and time. So we will go with this. Now the next question is question number 16. And question number 16 is, which statements are correct for a non-competitive inhibitor of enzyme action? So non-competitive inhibitor, which will not compete for the active site. So we have to focus on this. The first statement which they have given, the first statement is increasing the concentration of the enzyme substrate will reduce the effect. This is incorrect because this describes the competitive inhibition where the inhibitor and substrate fight for the same active site. So the inhibitor binds to an allosteric site or any other location. You can see that instead of active site, it can attach itself anywhere on the enzyme. So it will not affect, it will not reduce enzyme substrate, concentration of the enzyme substrate. So it will not affect. So this statement is not correct. The second statement, it can happen in that case when the competition is there is competition or competitive inhibition. So second statement is reduces the activation energy required for a reaction to take place. This is what an enzyme does, not an inhibitor. Inhibitor will not reduces the activation energy. This is the function of the inhibition. So third, it reduces the maximum rate of reaction. Yes, it reduces the rate of reaction because it is non-competitive inhibitor. So it will distort the shape of the enzyme. When the shape will be changed, so this makes some enzyme molecules effectively non-functional regardless of how much substrate is present. So the total number of working enzyme is reduced. So the reaction can never reach its original maximum speed. So statement number 3 is the correct statement. So according to this, we will go with the answer D. Now the next question, number 17, is which can increase the fluidity of the cell surface membrane at low temperature? Double bonds between carbon atoms in the fatty acid chains. Double bonds between carbon atoms, you can say those are unsaturated fats. So the double bonds create kinks in the fatty acid tails. These kinks push neighboring phospholipids away, preventing them from packing tightly. So this increase fluidity, it will not decrease, it will increase. So this statement is correct. Second is cholesterol. So this molecule acts as a temperature buffer. At low temperature, it gets it between the phospholipid tails and prevents them from crystallizing or packing together. So it will also increase the fluidity. Third one, fatty acids having shorter chains. Yes, fatty acids having shorter chains have less surface area, which means fewer hydrophobic interactions. So between them, so this makes the membrane less sticky and more mobile. So it will also increase the fluidity. So these three statements are correct. So question 17 has the correct option A. B, C, and D are not the correct options. Now we will move towards the question number 18, and question number 18 is, which statements about the proteins and glycoproteins in cell surface membrane are correct? They glycoproteins in cell surface membrane. So they can allow cells to bond together to form tissues. Bond together to form tissues. Proteins and glycoprotein act as cell adhesion molecules. So they will allow cells to stick to one another. So this is the correct statement. They can recognize messenger molecules like hormones. Yes, it is correct because many proteins and glycoproteins act as receptors. For example, an insulin receptor is a protein that waits for the insulin hormones to bind it. So this is the correct statement. So the third statement is, they can be antigens and allow cell-to-cell recognition. Yes, of course, it is. They can be the antigens. So glycoproteins with carbohydrates are the primary markers for cell identification. So this is how your immune system as a cell knows a cell is self rather than a foreign invader. So this is also the correct statement. The cell to cell recognition is allowed. So as these three statements are correct, so answer is A. Next question number 19 is, in an experiment, four different sized cubes of colorless agar were placed into a blue dye. The time taken for each cube to turn blue was recorded. Which show which row shows the surface area and volume of the cube that took the longest time to turn completely blue? You can see the surface area 6 and the volume is 1. At B is 9.2 and 2, and it 14.5 it is 3 and 30. So now we have to calculate the surface area volume ratio. So we will divide 6 by 1. It will be equal to 6. And at B, it is 9.5 divided by 2. It will be equal to 4.75. And on third, we will divide 12.5 by 3. So it is equal to 4.17, and then we will divide 38 by 16. So it is equal to 2.375. Now guys, see the surface area volume ratio is lowest at D. So D has the smallest surface area relative to its volume. So because it has the lowest surface, so then the die will take the most time to reach the center of the cube. So D is the correct option. Question number 20. The stalk of a dandelion flower is the hollow tube called a scape. Piece of these scapes are cut as shown by the dotted lines and be placed in surface solution, sucrose solutions of different water potentials. Which diagram shows the piece that is placed in the sucrose solution with the highest water potential. Highest water potential. So here you can see the inner cells and the outer cells. They have shown all these four pieces show the inner and outer. So, so if we talk about these, then thin-walled outer cells have are you can say they have sturdy, rigid cell walls that resist expansion. So they will not allow expansion. Whereas the thin-walled inner cells, these have flexible walls that allow the cells to expand significantly when they're taken in water. So if there is high water potential, as water will always move from its higher water potential towards the lower water potential. So when the plant tissues is placed in this solution, the water will move into the cells by osmosis and water potential to a lower water potential it will remove. So the inner cells having thin walls absorbs a lot of water and increase significantly in length. The outer cells having thick walls cannot expand nearly as much. You can see over here. So then it will also curl. So according to this, you can see when A is, in this case, A is the correct option because I have told you they have hard cells plus cell wall, so they expand less. So less water incorporated into it, whereas the inner cells they have. So this is the reason they they are they are they have absorbed lot of water because they have in the higher water potential. But in this case, you can see there is no reasonable chain in B and C when there is highest water potential. So, and whereas D, you can see, you can see this is the it has curled. It has curled, and the inner surface and the inner cells and the outer cells, they are, you can see, in this case, the outer cells has been shown that they are expanding instead of inner cells, whereas the inner cells should expand because they don't have the cell walls and the rigid cells inside. So D is also not correct. Only A is the correct option. Question number 21 is there, and question number 21 is telomers prevent the loss of genes during DNA replication, which now correctly shows cell types and the contain telomers. Typical animal cell is eukaryote, which have proper nucleus and chromosomes as well. Typical cell wall, plant cell also eukaryote. So it also has proper so telomers. Whereas typical bacterial cell, it has plasmid inside. So they don't need telomeres over here. So we will go with the answer B. Now question number 22, which events listed are part of mitosis? Part of mitosis. So they have given us three: interphase, prophase, and cytokinesis. Interphase, phase of the cell cycle where the cell grows and replicates its DNA before mitosis begins. So interphase is not part of mitosis. Prophase is the first phase of the mitosis. So it is part of the mitosis. Number three, cytokinesis. This is the division of the cytoplasm, not the, you can say, this is not a physical division of the because after this it takes place when there two nuclei are formed. So cytokinase is also not part of the mitosis. So according to this, only two is the part of the. So we will go with the answer D. Question number 23. Question number 23 is they have shown mitotic photomicrograph shows 17 cells in different stages of the mitotic cell cycle. They have cell one. Which row is correct for this photomicrograph? The role of a centromere in cell one. First of all, we will find out the role of the centromere in cell one. Attaches daughter chromosomes to spindle fibers. So this is centromere. Daughter chromosomes. So they attaches. So this is the correct statement. All sister chromosomes together. So the A is the proportion of the cell in the photomicrograph in telophase. The proportion in out of 70 cells, this is number 1, this is number 2, and this is number 3. These three cells show the same stage, of which is in the cell 1. So they are separating with one another and they are moving towards the opposite pole. So these are the three cells. So out of 17, three cells are there. If we find out their ratio, so the ratio will be less than, we will divide 17 with the 3. So in this case, we will divide 3 by 17, which is less than 2. So we will go with this. So according to this, A is the correct option. Question number 24 is the flow diagram shows some of the stages in the replication of a lagging strand of DNA. So here you can see which stage of DNA replication uses the enzyme DNA ligase. A, the DNA double helix begins to unwind. First of all, you can see the DNA ligase work as a glue. So it helps to bind, not to break the bonds. Whereas when the helical begins to unwind, there is breakdown of the, so A is not the correct option. Stage, the single strand of DNA are exposed again. This is not the C. DNA polymerase fragments are synthesized in the 5 to 6 direction. It will synthesize new strands. So this is not the correct option. The fragments of newly formed DNA are linked together. Now they will be bound, they will bind together with the help of this DNA like this. So D is the correct option. Because it catalyzes the formation of phosphodiester bonds to link these fragments together. into continuous strand. Now, question number 25 is, in a gene, the 20 common amino acids are each coded for by 3 bases in adjacent nucleotides. Why is there a difference between the number of amino acids and the number of possible triplet codes? The option, first of all, we must know that there are 4 DNA bases: adenine, thymine, cytosine, and guanine. Since they work in triplets, so there are 4 into 4 into 4, that is 64 options. So 64 possible codons. Triplet, you can say codons or triplet codes. So there are 20 different amino acids. The code is described as T generate or the abundance. So this means that some amino acids are coded for by more than one triplet. For example, the amino acid leucine is coded by 6 different triplets. Now, option A and C are 40 no functions. They have functions. So A and C are not some amino acids are coded by more than one triplet code. Yes. Some triplets have one known functions. They are start and stop triplets. So B is the correct option, which I have described. That is because there are 20 different amino acids, so they are used. They are using some amino acids are coded for about more than one triplet. So I have given example as well. All triplets have a known function. All they have known functions. So A, C, and D are not the correct option. Only B is the correct option because they go with the answer. Now, question number 26 is, how is a primary transcript modified to form messenger RNA? So, to turn up a primary transcript into functional messenger RNA, the cell undergoes a process called splicing. Thinking of it like film editing, you cut out the bloopers, scenes that do not belong to the final movie. So it becomes, you can say we have to remove some of the parts. So how is a primary transcript modified to form messenger RNA? It becomes longer as exons are joined together. Exons are the coding sequences. They are spliced together to form the final sequence. It becomes shorter as exons are removed. The exons are cut out and removed. Introns are non-coding. They are removed. So this is, it becomes longer as introns are joined together. Introns are not joined together. They are recycled by the cell after being removed. So they are not. It becomes shorter as introns are removed. Yes, this is the correct option. Now I would like to explain them again. Joining exons makes the messenger RNA functional, but it does not make it longer. It becomes functional, but then the primary transcript. So the transcript is already the maximum length. So this is not correct. If exons are removed, the cell would lose the actual instructions needed to build proteins. So this is not correct. It becomes longer as introns are joined together. Introns are not joined. They are recycled by the cell after being removed. So only B is the correct option, because in B, it becomes shorter as introns are removed. So introns are removed, so its size becomes shorter. Now the last of this video is the diagram shows protein synthesis in a prokaryote. You can see over here cell wall, DNA, messenger RNA, and what is X and what is Y, and then anticodon of transfer RNA1. So anticodon of this. So here you can see the molecule X is molecule X. You can see they have shown 4 triplet codes. So it means a polypeptide chain points to the chain of circle being formed. So these represent amino acid linked together. So while a completed unfolded version is protein, the growing chain during translation is specifically called a, you can see, we will call it a polypeptide. So X is a polypeptide. Then Y, you can see it is the unit of the ribosome, which is 70S. The question explicitly states that this is the protein synthesis in a prokaryote. So prokaryotes like bacteria have 70S ribosomes, eukaryotes have 80S ribosomes. Therefore, structure Y must be the 70S. Now, anticodon of transfer RNA, you know, which is uracil, and guanine. So the anticodon will be adenine, and cytosine, because uracil always pairs with adenine and guanine always pairs with cytosine. So according to this, we will go X is polypeptide and 70S chromosome, adenine, and cytosine. So C is the correct option. So it was all about the second video from question number 15 to 27. Please like, share, and subscribe my channel to watch more videos. Thanks for watching, and wait for third part of this paper. Thank you.